of the Medical Society of DC. I'm here to give you a couple of housekeeping tips and tricks for tonight's session. First and foremost, you'll notice that you have come in on mute in tonight's program. That is on purpose. While I'm sure everyone has much to share, we prefer that you share it in the chat box. We are using Zoom, so hopefully everybody can find the chat box at this point. Please enter your questions and comments there, and our speakers will handle everything from the chat box. If for some reason you can't find the chat box, feel free to email me directly. It's hey, Hay, at msdc.org, and I can relay the questions to the speakers. Tonight's session will be recorded. That is to allow people who were unable to attend to view at a later date. So while we do not share that our sessions are off the record because they are recorded, we do ask, of course, that you maintain a general level of respect and don't go calling your favorite Washington Post reporter as the session is going on. Finally, I encourage everybody to take a look at our calendar of events for upcoming events. For those who are unfamiliar, Washington Healthcare on Wednesdays is a weekly content series of events, some pre-recorded and some live. So in February, we've already started scheduling some very exciting events, one that is definitely up on the calendar and some that will be soon on the calendar. So please plan on clearing your Wednesday schedules after 5 o'clock. So without further ado, I'm going to turn the microphone over to Dr. Desiree Pineda, immediate past president of the Medical Society of DC and member of the Mayor's Vaccine Scientific Committee. So Dr. Pineda, the floor is yours. Thank you.
Thank you, Robert. Today we have distinguished guests, and this is really important now with all the vaccines going on. A lot of us have already been vaccinated, but a lot of them more coming into the file. So, and not only scientifically, but politically, it's a very important topic. We're gonna leave the politics to the side, but I'm going to introduce Dr. Marc Siegel. He's an associate professor of medicine in the Division of Infectious Diseases at GW, University School of Medicine and Health Sciences. He obtained his medical degree from Georgetown University and completed his internal medicine training at Georgetown University Medical Center. Then he went to New York and ran his own internal medicine practice in York. So then he went back here to DC in 2007 to pursue his fellowship in infectious diseases. He is a co-investigator at GW for the Cov study, looking for the efficacy of the Moderna mRNA vaccine against COVID-19 or SARS-CoV-2 virus, as well as the principal investigator for an investigational monoclonal antibody to interfere with the inflammatory cascade in patients hospitalized with severe COVID-19. His initial research was focused and centered on chronic hepatitis C, but with the advent of the effective hepatitis C therapy, now he's doing research in HIV pre-exposure prophylaxis, or PrEP, and he has begun the study of the as a medical lead investigator or principal investigator on not only 1, but 7 PrEP trials, and funded through the HIV Prevention Trials Network. So Dr. Siegel, the floor is yours.
Thank you, I appreciate it. I am gonna try and share my screen. So let me make sure that everybody hopefully can see this. And let me start the slideshow. So can you, Dr. Pineda, can you see this?
Yes.
Okay, great. All right, so we're going to review the COVID-19 vaccines and talk a little bit more generally about the rollout and some other pertinent topics that are current. So plan to start talking about the platforms, the different types of vaccines, the results from the trials that we already have results from, and then talk a little bit about the distribution and administration and some of the roadblocks that we've seen. So my disclosures, I am one of the co-investigators for the Moderna vaccine study that we're doing here at GW. We will also soon be doing the Sanofi vaccine study as well. I am not a vaccinologist, so you know, my knowledge of all things vaccine is more limited than someone who has a career in vaccines, but I'm learning quickly all things vaccines, and I have no conflicts of interest. So let me just minimize this. So I'm sure you're all up to aware of the current status of the pandemic in the US. 25 million cases, 420,000 deaths. In the healthcare field, we've had about 380,000 healthcare workers get infected, and unfortunately, we've lost about 1,300 healthcare workers. And even though we may have peaked from our post-Thanksgiving peak, we are still at very high levels, and with around 100,000, close to 200,000 cases a day, and 3,000 to 4,000 deaths a day. So as far as the coronavirus, the SARS-CoV-2 virus, it has 4 major proteins, and the protein that we have and all the companies have decided to target for vaccine development has been the spike protein, which is on the surface and has 2 conformations, has a pre-fusion conformation, and then once it fuses with the ACE receptor in the lungs, which it binds to gain entry into our respiratory cells, it changes the conformation slightly. And that's just important because all the vaccines have targeted the pre-fusion state of the conformation of the protein. And once it binds to the ACE receptor, it gets endocytosed, and then it gets cleaved from the membrane, and then fuses with the endosome, and then releases the mRNA into the cells, and then that causes the active infection. So as far as different vaccine candidates, we have 4 different broad types. There's the virus candidates, where we use actually either an inactivated or attenuated version of the coronavirus. None of these are being pursued in the US, but they have been pursued in other countries, including China. We have the nucleic acid platforms, which is the mRNA vaccines that are currently approved. There's viral vector platforms, which use either attenuated or inactivated viruses to transport the RNA or DNA into our cells. And then we have the more standard protein-based vaccines, which is kind of how most vaccines have been produced up till now. So before we talk more about the different types of vaccines, I think it's also important to understand a little bit about Operation Warp Speed, because it's quite important in the development and the rollout of these vaccines. So this was an initiative that got started initially in April and then got officially announced in May 15th of 2020, which was a public-private partnership between the Department of Health and Human Services, CDC, NIH, BARDA, and the Department of Defense, with private industries to help accelerate the development, manufacturing, and distribution of the COVID-19 vaccine and other therapeutics like the monoclonal antibodies, and also to help in the development of diagnostic tests for the SARS-CoV-2 virus. It also was responsible for starting the manufacturing process of these vaccines and therapeutics at industrial scales before we even knew that there was any efficacy, which is ultimately highly important for being able to get to a point like we are right now, where within less than a year of starting the development of the vaccine, we already have vaccines being distributed. Its initial goal was actually to produce and deliver 300 million doses of safe and effective vaccines starting in January of 2021. So they actually beat that goal by starting the delivery of these vaccines in December. Initial funding came from the CARES Act, and the initial funding was $10 billion, but that had to be increased in October 2020 to $18 billion to cover extra expenses. So if you look at the vaccine candidates that Operation Warp Speed has supported, it's given about $9 billion to 6 companies: Pfizer, Moderna, AstraZeneca, Janssen, Novavax, and Sanofi. And these are all, so they're all run on different, or they don't all run on different platforms. 2 of them are mRNA vaccines, 2 are viral vaccine vectors, and 2 are protein-based vaccines. And for those, we have commitments of different amounts. So Pfizer's initial commitment was for 100 million doses for that amount, with an option for the US government to purchase more. Moderna also was a contract for 100 million doses. AstraZeneca contracted for 300 million doses. Janssen for 100 million, Novavax for 100 million, and Sanofi for 100 million. So we have obviously probably going to exceed that, purchase more than those amounts that we initially contracted for. And as long as the supply is available, I think the government will continue purchasing vaccines to try and get everybody in the US vaccinated. Operation Warp Speed actually funds other things outside of just vaccines. It funds basically everything to do with vaccine development and also distribution. So in May, it actually made a contract with a company called Appijet to produce more than 100 million pre-filled syringes by the end of 2020 and another 500 million in 2021. In June, they made 2 contracts with Corning and with a company called SIO2 to produce the glass vials that are needed to actually put the vaccine in before transportation. In August, it contracted with McKesson to be the sole distributor of the Pfizer vaccine, which needs ultra cold storage. Sorry, not of the Pfizer vaccine, that's my mistake, of the Moderna vaccine, which needs cold storage, but not the ultra cold storage. And then in October, it actually created this pharmacy partnership for long-term care program, which was an agreement between CVS and Walgreens to facilitate vaccinations of long-term care facility residents. And this included everything, including the cold chain management, the vaccinations on site, the reporting requirements, all at no cost to the facilities or the residents of these facilities. So one of the things that Operation Warp Speed allowed us to do and actually encourage was, accelerating the development of vaccines. So historically, vaccines have taken 5 to 10 years to develop because usually it's a sequential process where you first have to identify the target and decide which is a right target. So is it a spike protein? Is it a nuclear protein? And then you obviously start with a phase 1, which are the first in human trials and dose escalation trials, and then the early phase 2, where you're doing the safety and early efficacy trials. And then usually, once you have the data from those, you will move into the larger phase 3 trials, which are, the 30,000 patient trials. And it's only usually by the time you're in the phase 3 trials and you're starting to see relatively good efficacy data that the company would then start the manufacturing scale up, because obviously they don't want to risk putting money into manufacturing and then having a vaccine that's proven ineffective and they've wasted all that money. What Operation Warp Speed did was allow us to condense that by overlapping a lot of those processes. So they overlapped the phase 1, phase 2, and phase 3. So we didn't wait for the phase 1 to be finished before we started phase 2, and we didn't wait for the phase 2 to start before we started phase 3. But what we did was we waited for enough data to know that we could safely proceed. So for example, in the Moderna vaccine study, we didn't start the phase 3 trial until everybody in the phase 2 trial had their first dose of vaccine and had at least had 28 days of safety data. So we knew that in the first month, there was no safety concern. So we could safely start the phase 3 trial with the first dose of the vaccine, knowing that there was enough safety data to do that. And that basically condensed the whole process substantially. The other thing that the Operation Warp Speed did is started the manufacturing process right at the beginning. So rather than wait for early efficacy data in the phase 2 trial, we were starting the manufacturing process, which was a risk because obviously if the vaccines had failed and had been proven ineffective, then all that manufacturing would have been for naught and all the vaccine that would have been produced would have been discarded. But luckily that ended up not being the case. And actually one of the first vaccines that had this similar platform was the polio vaccine in the 1950s and 1960s. And that was at that point funded by the March of Dimes and was highly effective in getting a vaccine out quickly when we had such a dire need. So the first vaccine platform is the mRNA vaccine, and this is the Moderna and the Pfizer vaccines. And these are basically taking a piece of genetic code, either RNA or DNA, and inserting it into this lipid nanoparticle, and then injecting that. It gets taken up by the cells, and in the cells, we use the host ribosomes to produce the protein that then gets taken up by what is called our antigen-presenting cells. And those antigen-presenting cells will then chew up that protein and put the piece of the protein on the surface, and that then elicits our humoral or antibody response and a cellular or T cell response. So the challenges historically have been that mRNA is very unstable. And so historically, we haven't been successful because usually the mRNA gets broken down too quickly to induce an immune response. It's also had unintended inflammatory responses in patients, and up till now has only had modest immune responses. The benefits are we're not injecting anything that's infectious. We're injecting just a tiny bit of code for one part of the virus. And it actually is very easy to produce this mRNA, and these manufacturing platforms had already been established. So the time to produce this mRNA and produce the actual vaccine is very short. We have recently developed this technology where we've been able to envelop the mRNA in this lipid nanoparticle, and that actually allowed the mRNA not to be degraded by the host cellular enzymes and allowed the uptake into the cells where the ribosomes could then produce the protein. And so that was one of the advances that has made it successful now, where historically it has not been successful. So the first two of the mRNA vaccine candidates that went into phase 3 was a Pfizer, and this started in July and was looking at patients over the age of 16. It was a randomized placebo-controlled study and 2 doses given 21 days apart. And it was looking at basically symptomatic COVID 7 days after the second dose of the vaccine. The secondary endpoint was looking at people who actually got severe COVID disease on the study. It enrolled about 44,000 patients. Half of them were female. Median age was 51. Had about 26% Hispanic, 10% Black, and 4% Asians, and had about 35% of population that had a risk. One of the major risk factors for severe COVID, which was obesity. So its study results came out in November, and it met its primary endpoint when they'd had 162 patients in the placebo arm who developed symptomatic COVID and only 8 in the vaccine arm, which gave it a vaccine efficacy after 2 doses of 95%. It also could tell that there was some efficacy after just 1 dose, and after 1 dose, at the time that the person got their second dose of the vaccine, there was about a 52% vaccine efficacy at that point. And these efficacy results were consistent regardless of age, gender, race, or weight. When we looked at the secondary endpoint of severe COVID disease, there were 4 cases total, 3 in the placebo group and 1 in the vaccine group. So the efficacy against severe COVID was 64%. And this Kaplan-Meier curve here, you can just see that roughly at day 12, you start seeing the divergence of the placebo group, which is a red line, versus the placebo group. So there was a divergence between the vaccine group, which was red line, from the placebo group, which was the blue line, showing that there was already efficacy way before the endpoint, which was 7 days after the second dose. So safety was relatively safe. There were some localized side effects. Injection site pain was the most common, fatigue, headache. There were some reports of fever that were more common after the second dose than the first dose. About 16% of people reported fever. There were about 8.8% of people who had a grade 3 adverse events, the most common being fatigue and headache. Surprisingly, older patients had fewer of the side effects and had milder adverse effects than the younger population, probably to do with a lot of these side effects are due to the robust immune response to the vaccine. And so in early December, the FDA granted the emergency use authorization for this for anybody over the age of 16. Moderna started its study at the same time. This is also an mRNA platform. It had patients over the age of 18. The only difference here, the doses were 28 days apart, had basically the same primary and secondary endpoints looking at symptomatic COVID disease. And here they define symptomatic COVID as having a positive nasal swab for SARS-CoV-2, plus either shortness of breath, cough, or pneumonia on X-ray, or 2 of fevers, chills, myalgias, headaches, sore throat. They enrolled 30,000 patients. Their demographics were roughly the same. About half were women. Age was roughly the same at 51. Demographic makeup was roughly the same, 20% Hispanic, 10% Black. We did have 25% healthcare workers and 22% with at least 1 high-risk condition for severe COVID. And their results were surprisingly similar to the Pfizer. So they had 185 cases in the placebo arm, 11 in the vaccine arm, giving a vaccine efficacy of 94%. They had a higher vaccine efficacy after the first dose. So their vaccine efficacy after the first dose was 80%. Remember, Pfizer's was only 52%. But one of the reasons is they had 4 weeks between the first 2 doses versus Pfizer's 3 weeks. So Pfizer, presumably if there'd been a longer time between the first and second dose, would have seen a higher vaccine efficacy just after the first dose as well. Their secondary endpoint of efficacy against severe disease, they had 30 cases of severe disease, and all of these were in the placebo group. 9 required hospitalization, and no one in the vaccine group who developed symptomatic COVID developed severe disease, i.e. requiring hospitalization. And the Kaplan-Meier curves on the top, you can see it as per protocol, which started 2 weeks after the second dose, which is where the hashed line is. On the modified intention to treat is all comers starting after the first dose. You can also see the divergence of the line, the Kaplan-Meier curves, roughly at the same point, roughly at day 12, you start seeing this divergence, showing that you do get vaccine efficacy after a single dose. The side effects, almost similar to the Pfizer. So most people had some injection site pain, fatigue, headache, myalgias. Similar, 15% had fever after the second dose. There were some grade 3 events, less grade 3 events after the second dose than the Pfizer, also less common in people over the age of 65. There was some hypersensitivity reaction. All of them were deemed unrelated to the mRNA vaccine. There were 4 cases of Bell's palsy, 3 in the vaccine group, 1 in placebo group. And this in analysis was felt to be consistent with background incidence of Bell's palsy. It's not studied in pregnancy, and pregnant women were excluded, but somehow 13 people either did not get pregnancy tested or developed pregnancies during the trial. 7 were on the vaccine arm, 1 on the placebo arm. There was 1 spontaneous abortion and 1 termination, 1 in each arm of the study. And obviously we don't have any outcomes data yet. So this got granted the EUA by the FDA 1 week after the Pfizer on December 18th. So the second platform is the adenovirus vector-based vaccine. And so this is when you take either some kind of viral vector. So usually we use adenovirus, but you can use other viruses such as a weakened measles virus. And you basically take out its cellular machinery and you insert the genetic machinery that you want. So you can insert the DNA or the RNA that you want the virus to insert into the cells. And the benefits are it's relatively easy to manipulate the genetics of the cellular machinery of these viruses. They do induce a very robust T cell and antibody response. We have this process established, so it's something that we've been doing for a while, and so it can be produced at large scale. Unfortunately, some of the disadvantages is it's more time consuming than the mRNA vaccines, and it does require an annual booster shot as well. So the first of the adenovirus vector vaccines is the AstraZeneca study. And so this is using a chimpanzee adenovirus vector. And why chimpanzee? Well, because you don't actually want to have people who already have immunity to this adenovirus vector, because otherwise their antibodies will neutralize the vector before it can actually insert the RNA or DNA into the cells, where it will then be produced into the spike protein. So they use a chimpanzee adenovirus because humans presumably don't have any immunity against that. Its phase 3 study started in the US in August. It is fully enrolled, and we expect to see the studies in the next couple of weeks. It is 2 doses, 28 days apart, randomized on a 2-to-1 ratio and placebo controlled. We do have some preliminary results from studies that were carried out in the UK and Brazil. And in the UK, they actually had a little bit of a mess up in their study. So they started studying in the UK, giving a half dose. That was not what the protocol had written. And when they found this, they paused the study and then restarted it with the correct dose a month later. In that group, they saw a vaccine efficacy of 90%. So 30 people in the placebo arm ended up getting symptomatic COVID, 3 in the vaccine arm. They did have another 9,000 participants who got 2 full doses a month apart as the protocol was written. And in that group, they only saw 62% vaccine efficacy, which seemed strange that, that the 2 full doses would have a lower vaccine efficacy. And we'll talk about that in a second. And when they combined the numbers and looked at the total 10,500 patients, they saw, or 11,500 patients, they saw a combined vaccine efficacy of 70%. It was statistically significant. And on the basis of that, it has been approved in England and several other countries in Europe as well. So if you look, you can see that the vaccine efficacy overall was 70%. And you can see, again, looking at just the cases of severe COVID, there were only 6 cases. They were all in the placebo arm, none of them in the vaccine arm, which suggests that even if it didn't prevent people from getting symptomatic COVID as well as the mRNA vaccines, it still prevented people from coming to the hospital, which is ultimately probably the most important thing about these vaccines, because we want to take off the burden of our healthcare system. And it's the people that actually get admitted to the hospital that are eventually going to have the risk of having a fatal outcome. So, you know, even though 70% is obviously not as enticing as 95%, it still, as a secondary endpoint, still achieves that secondary endpoint. So when we looked at why the results were better for the people that got the half dose versus the full dose. So remember, some of those people, probably the people who got the half dose, when the study got paused, they didn't get their second dose until much later than was originally planned. And some people didn't get their second dose until more than 12 weeks after the first dose. Not what the protocol intended, but just what happened because of the pause. So when they compared the people in that first, the 2,000 people who got the half dose followed by the full dose, and looked at the antibody levels, they noticed that people who got it, the two doses less than 6 weeks apart, had an antibody level that was a third of the antibody level in the people that got the doses 12 weeks apart. So this is still trying to be sussed out and and the scientists are still trying to figure out what this means. But on the basis of this is one of the reasons why in England they've pushed out the second dose of the vaccine to 12 weeks from the first dose, because they believe that by waiting, you may actually generate a higher immune response than by giving the vaccines 4 weeks apart. Obviously, we don't have the data, and England's doing a real-time study to see if this actually works. But it was just an interesting outcome from this study. So the other adenovirus-based vaccine that's out there is by Johnson or Janssen, and they're using a replication-deficient adeno-26 viral vector. So adeno-26 is not a common human pathogen. It can become pathogenic, but it's very uncommon. And so most people don't have pre-existing immunity. So again, we won't neutralize this virus before it actually inserts the RNA into our cells. It has been looked at before for Ebola, Zika, RSV, and HIV, has not actually been successful in any of those. There are 2 different phase 3 studies going on right now. There's the Ensemble study, which is the single dose given to 45,000 patients in a 2-to-1 ratio. It is fully enrolled, and we're expecting the results either this week or next week. And so we're hoping that maybe by the end of February, if the results are positive, and the word on the street is the results look good and will be quite impressive, that we might get this approved by the FDA and then the EUA before the end of February. There is actually a second study called Ensemble 2, where they are looking at 2 doses, 57 days apart. It was launched in November. It's still enrolling, and I'm not sure when the results of that will become available. So the last platform is the protein-based vaccine, and this is kind of historically what we have done. And this is where we take multiple or a single or multiple fragments of an organism, and they're either cleaved or purified from the organism, or they're expressed in recombinant form. So we actually generate them in a lab. And so the advantage of this is we know how to do this. We've been doing these kind of vaccines for a while. We can produce it at large scale. There's good reproducibility between batches, and we have established safety for these types of vaccines. The disadvantage is they generally aren't as immunogenic by themselves, so you need to give them with an adjuvant, and you do need to give booster vaccines year after year. And so those are some of the downsides. The two vaccines that are using this platform are the Novavax. And so they have the SARS-CoV-2 RS vaccine, which is using a matrix M1 adjuvant. And the adjuvant basically increases the immune response to the protein. And so this is a study that has been going on in the UK since September. It's enrolled 15,000 patients. It's fully enrolled. And the study in the US was delayed quite a bit. It only started in December, late December. It's already enrolled more than 10,000 patients. That was actually data from a week ago. Its goal is to enroll 30,000 patients. It's going to be giving 2 doses 21 days apart. And so we probably won't have the data from this for at least another month and a half. Sanofi is the other protein-based vaccine. It's also using an adjuvanted recombinant pre-fusion spike protein. So we talked about the pre-fusion, the post-fusion state. It had an initial phase 1/2 study that started in September, looked at 420 patients, looking at the two different doses of the antigen, and looked at 2 different adjuvants with that. Unfortunately, what it found is that even with a higher dose of the antigen, there was actually a quite poor immune response, and the immune response was poorer as people got older. So in patients over the age of 60, only 62% actually developed any kind of an immune response. So they basically had to go back to the drawing board and pick out a new antigen. And so they're now starting a new phase 2 study. It's going to be starting here at GW as one of the sites. And they're going to enroll 720 patients in 1 week across the US. And they're going to give them 3 different doses of this new antigen with the adjuvant that they decided was the better of the 2 adjuvants. And so hopefully on the basis of that, they'll decide which dose to use and then move forward with a phase 3. But you know, the problem with this is now it's getting very difficult to enroll, especially people over the age of 65, since most people over the age of 65 are now eligible for the vaccine under the EUA. And it's also becoming more problematic about whether they can still do a placebo-controlled trial, given that it may be unethical to offer people placebo if they have access to a EUA vaccine. And so the question will be, can they do a placebo-controlled, or will they have to do it based on neutralizing antibody levels? And so those are questions that have yet to be answered. So as far as logistics of getting the vaccine out, obviously the Pfizer and Moderna vaccine have some logistics. The Pfizer is the one that requires ultra cold storage. So it's produced in Kalamazoo, Michigan, and it's being distributed to 1,000 sites in the US. It has to be stored at -70. So Pfizer has this huge warehouse with a whole bunch of -70 freezers. And at -70, it can be stored for 6 months. It's shipped in these special thermal shippers, which have GPS-enabled temperature controls, so we can monitor the temperature. And in these shippers, it can be maintained at -70 for up to 10 days. Each shipper can hold 5,000 vials, and each vial can hold technically 5 doses, but we're finding out that maybe some of the vials can hold 6 or 7 doses, depending on the type of syringe you have to get get all of the liquid out of the vial. It does require thawing, and once it's thawed, it requires dilution. So it's a little bit of a 2-step process. And then once it's diluted, you can keep it in the fridge at the fridge temperature for 5 days. But if it's thawed to room temperature, it's only stable for 2 hours. And so it has to be administered fairly quickly after that point. Moderna is similar, but has a slightly lower cold storage requirement. So it's only required to be stored at -20 degrees. It's being produced at the primary Moderna vaccine production facility, which is in Norwood, Mass. But it has outsourced to 2 other production facilities, one in Bloomington, Indiana, and one in Portsmouth, New Hampshire. And this is the one that's being distributed by McKesson under Operation Warp Speed to 3,400 locations throughout the US. Similar to Pfizer, it's stable at 6 months at -20. Each vial contains 6 doses. It does need to be thawed, but does not need to be diluted. It can be stable in a refrigerated temperature for 30 days, but once thawed, has to be used within 8 to 12 hours. So here's kind of the Operation Warp Speed distribution process. So you can see Pfizer at the top. Pfizer's vaccine goes directly via UPS or FedEx to their distribution centers, and then FedEx and UPS actually distribute it to the 1,000 distribution centers throughout the US. Pfizer and UPS actually have also bought 2 production facilities to produce extra dry ice, and those are at their hubs in I can't remember where the hubs are. The Moderna is at the bottom, and the Moderna vaccine goes to McKesson distributors, who actually also then distributes all the kitting facilities, so the syringes and anything else needed to distribute the vaccine. And then they will actually distribute it either through their own distribution network, or they will also use FedEx and UPS to get it to the 3,400 distribution centers. So the other vaccines that haven't been approved yet are actually much easier to distribute because none of those require ultra cold storage. So all the other 4 vaccines just require refrigerated temperature for storage. And so logistically, those will be much easier and will probably allow for expansion of distribution centers, pharmacies, and other centers that aren't necessarily distributing the vaccine right now. So when the CDC initially proposed how the vaccines should be distributed, they had phases, phase 1A, phase 1B, phase 1C. So if the phase 1A was initially healthcare workers and residents of long-term care facility, that was estimated to be 24 million people. Phase 1B was then going to be essential frontline workers and people over the age of 75, which is going to be another 49 million, followed by phase 1C, patients between the age of 65 and 74, and then people with underlying medical conditions and other essential workers. So those first three phases were going to be 200 million people. And obviously states have had the leeway to decide whether to do this sequentially or do some overlap. I think most states have decided that rather than wait for every person who meets criteria for phase 1A to get vaccinated, once the initial surge of the phase 1A was met, even if not every healthcare worker had been vaccinated, they opened it up to people in the phase 1B category and even made tiers in each of these categories. So phase 1B, tier 1 or tier 1B. So every state has kind of done this a little differently. Some states started with people over the age of 65 right away. And so there has been some heterogeneity across the country in who's been eligible. If you look at how we're doing, we had a slow start. We started in late December with the distribution. There were some hiccups. There were some snowstorms that delayed the distribution of the vaccine. So we have actually done a lot better recently, and I think we are now regularly administering over 1 million doses of the vaccine a day. So of the 47 million doses that have been distributed, over half have actually been administered, and about 4 million people have actually also received fully 2 doses of the vaccine. This does not include the data for long-term care facilities. If you look at the makeup of the 2 vaccines, so Pfizer was actually, got the EUA a week before Moderna, so they've had more doses administered, 13 million of the Pfizer versus 11 million of the Moderna. And if you look at the map of where the most vaccines are being distributed, this is not quite up to date because DC is actually currently the jurisdiction with the highest distribution rate, and we'll get to that in a second. But West Virginia has done very well. Unfortunately, some of the southern states have done poorly. Georgia has been one of the least effective state in getting vaccines distributed. So generally, the country has been a little bit of a hodgepodge, but I think we are figuring out where the roadblocks and barriers have been, and we are picking up pace. I think the goal is now to get to at least 1.5 million a day. And most experts say that if we really hope to get ahead of this pandemic and actually get to herd immunity, we should be aiming for somewhere between 2 and 3 million doses of vaccine a day. That may not be possible because the supply may not be able to meet that demand. But obviously the administration is doing quite a bit to try and speed up the production of the vaccine as well. Looking at DC's vaccination data, this is publicly available on the DC coronavirus vaccination page. So this was updated on Monday. So we've administered 75% of the 68,000 doses of vaccine that we have received. Now, DC unfortunately did not get vaccine distributed based on our workforce. We got it distributed based on our population, and the DC Department of Health knew that was going to be an issue because we have so many people that work in DC that live in other jurisdictions. But DC decided, despite that, that they would not restrict giving vaccine to just DC residents, especially in the first wave where it was healthcare workers. And so plenty of the people got vaccine. About 50% of the vaccine doses administered so far have been to non-DC residents because of that. Luckily, Virginia and Maryland have recognized that and have given some of their allocations back to DC to compensate for that. If you look at the distributions by ward and what percentage of residents over the age of 65 have actually received their first dose, the majority or the high percentage is in wards 2 and 3, and lower percentage in ward 7. And DC has been acutely aware of equity in the vaccine distribution, and as of week 6, actually when they had new vaccine that was going to become available, restricted it for 48 hours to people from the wards that had a higher rate of death related to COVID, so wards 5, 7, and 8, and ward 1 as well. And then only after 48 hours, so they actually took 75% of the total allocation and allowed those wards which are more severely affected to have first access to that, and then after 48 hours would open up the other 25% of the allocation and let anybody in any of the wards in DC make an appointment for that. If you look at the appointments, what percentage of all the appointments have been made by different wards, you can see that Ward 8 still is at the lowest end, and Ward 7 as well. One of the things that to point out is if you look at people who are eligible, Ward 1 and Ward 8 have the least number of people that have been eligible up to now. So they have the lowest number of healthcare workers who were the first to be eligible, and they also have lower proportions of patients over the age of 65. So you have to read that data knowing that, you know, it's not just because there's inequity in accessing, you know, computers to be able to make the appointments, or maybe there's not the literacy with using computers. There is also some element of who was actually eligible living in those wards. DC actually at the beginning knew they were going to have to understand the population and decide and try and message how to make sure that the majority of the DC population wanted to get vaccinated or would feel safe getting vaccinated. So they did a survey of 6,500 people in November and December, looking at people who identified as a certain race and whether they were willing to get the vaccine. And they noticed while over 90% of Asians and whites said they were willing to get the vaccines, only 60% of people identified as Black were willing to get the vaccine. And then when they looked based on employment status, you know, even though they saw that, you know, healthcare workers by the majority were willing to get it, there were still 20% of healthcare workers and 25% of essential workers who said they were not willing to get the vaccine. When they delved a little further and looked at race and then essential workers and healthcare workers, you know, 45% of essential workers identified as Black and 40% of healthcare workers identified as Black said at the time the survey was done, which granted was almost 2 months ago, said at that point they were not willing to get the vaccine. And why? The number one was safety concerns about the safety and side effects. And the second was because of the speed that the vaccine was made. Now, I think this is probably, if you did the survey again today, you probably would see a difference. You would probably still see a disparity between people identified as Black and people identified as white, as Asian. But I don't think the disparity would be as wide, because now that we've seen that it is a relatively safe vaccine and that the administration is going relatively well, I think people's opinion about the safety of the vaccine and their willingness to get the vaccine hopefully has changed. So this is also available data on the DC coronavirus vaccine website. So this looks at all 51,000 doses that have been administered up till now. The top bar shows DC residents, 25,000. The second bar, non-DC resident, 24,000. So pretty equally distributed. I am a Bethesda, I'm a Montgomery County resident. I got my vaccine here in DC because I work in DC. And so, you know, DC was very gracious in that they allowed people who did not live in DC to get their vaccines here. So obviously everybody's heard about these anaphylactoid reactions that have happened. They happened initially in England with 2 nurses, and then they happened fairly quickly after that as we started distributing the Pfizer vaccine. The CDC has put out 2 MMWRs, one based on the cases for the Pfizer vaccine through from December 14th through the 23rd. They reported 21 cases out of nearly 2 million doses administered, which was a rate of 11 cases per million doses, which is higher than what you expect to see with most vaccines. So the flu vaccine has a rate of anaphylactoid reaction about 1 case per million. So we definitely noticed it was higher, but when you looked at the cases, the majority of these people were people that did have a history of allergy or allergic reaction. 7 had a history of previous anaphylaxis, and the majority of these vaccinations occurred within 15 minutes of vaccination. When they looked at the Moderna, they saw, then this was looking from December 21st through January 10th, there were 10 reported cases of anaphylaxis out of 4 million doses administered. So they seem to have a low rate of anaphylactoid reactions, 2.5 cases per million doses administered. And again, most of these cases, 9 of the 10 had a history of allergic reactions, and 90% of these cases occurred within 15 minutes of the vaccination. The current thinking is that the reaction is due to a reaction to polyethylene glycol, And polyethylene glycol is used in medications, and we do know that people can have allergic reactions to this. These are the first vaccines to use polyethylene glycol, and it's a stabilizer of that lipid nanoparticle we talked about that stabilizes stops the mRNA from getting degraded. And the current thinking is people with high levels of antibodies, high levels of IgG to polyethylene glycol are more likely to react. So guidance is that if you've had a history of reactions to polyethylene glycol or polysorbate, which is similar to polyethylene glycol, it's not used in the vaccines, but it's used in medications, that's one reason to not get vaccinated. So current CDC recommendations are, if you have a severe allergic reaction to the first dose, don't get the second. If you have a non-severe allergic reaction, but it's within 4 hours, you should probably not get the second dose. If you have had an allergic reaction to any other type of vaccine, so if you've had an allergic reaction to the flu vaccine or the shingles vaccine, you probably can get the vaccine, but you probably should consult with your physician, even though there's no solid data. It's basically the participant's choice if they choose to get it. If you've had allergic reactions but they're not related to vaccine, it is safe to get vaccinated, but we will observe you for 30 minutes rather than the standard 15 minutes because the majority of these reactions do happen within 15 minutes. If you've ever had an allergic reaction to polyethylene glycol or polysorbate, you should not get either of these two vaccines. The other vaccines that are coming out, so the AstraZeneca vaccine and the Johnson & Johnson vaccine will not have polyethylene glycol in it. So presumably patients who cannot get these mRNA vaccines because of a history of allergic reactions will probably be recommended to get those vaccines. Herd immunity comes up a lot because this is ultimately what we hope we will attain if the virus doesn't mutate too much. Just so you know, even though the former advisor to the White House, Dr. Atlas, was recommending that we quarantine our elderly and let our healthier, younger population get naturally infected and develop natural immunity as a goal towards herd immunity, that has never been a way of getting herd immunity. Herd immunity has always been a goal of vaccination campaigns. So herd immunity with measles vaccination or with smallpox vaccination, but we don't want herd immunity through natural immunity because that means you're going to basically expect to have a certain number of patients die in the quest to get natural herd immunity. How do you calculate herd immunity? Well, it's a calculation that's pretty simple. It's 1 minus 1 over the R naught. And what is the R naught? The R naught is how many people, if you put an infected person in a room with uninfected people, how many people you would reasonably expect that one infected person to infect. Current estimates suggest that the R naught of this virus is in the range of 2 to 3. Just so you know, the R naught of the regular seasonal flu is about 0.1, and the R naught of the swine flu from 2008, 2009 was around 1, maybe a little higher. So this is clearly more infectious than either of those. The R naught of measles, which is the most infectious virus that we know, was around 15. And this diagram shows that the higher the R naught, if you put that number in here, the higher the threshold, the higher the proportion of the population that needs to get immune before we develop herd immunity. So with an R naught of between 2 to 3, the current estimates are that somewhere between 50 and 66% of the US population will need to have either natural immunity or immunity from vaccination before we could start seeing some form of herd immunity. But that will not happen right away. That will slowly tail off. Obviously, in the news are the new COVID variants. So the first COVID variant was discovered in the UK, and it's termed the B117 variant. I know that the easier term is the UK variant, but similar to trying not to name pandemics after the country where they were first identified, we are trying to not name these variants after the country where they first sequenced, because they very likely didn't start in the country. And you don't want to stigmatize a country for doing what is actually very important work, which is surveillance. And the UK had a very robust surveillance and they were sequencing up to 10% of all the isolates, where the US was sequencing less than 1% of isolates. So this was discovered in the UK, but we shouldn't permit the UK variant, because otherwise that might be stigmatizing against the UK. But this was the first variant that was discovered, and it had 8 mutations. It actually had 23 total mutations, but 8 mutations in the receptor binding domain of the spike protein, which was a concerning part of this. And one of the things that made the virus, the spike protein, attached to the ACE receptor with greater affinity, and therefore made it more infectious. Since then, there have been at least 2 other variants. So there's been the B1351 variant, which was first identified in South Africa, and the B11248 variant that has recently been identified in Brazil. They all have mutations in the receptor binding domain of the spike protein. And they all have this mutation in the N501Y nucleotide, which is responsible for this increased affinity for the ACE receptor. We think that this has increased the R0 of these viruses by somewhere between 0.4 and 0.7. And why is that important? Well, if we go back to our previous slide, that means more of the population will need to be immune before we might see herd immunity. Now, unfortunately, the first, when these variants first were like hitting the news, everybody said, well, they're more infectious, but they don't seem to be causing more severe disease or more mortality. Recent studies have shown, there was a report out of England about 5 different studies that suggested that it has increased the mortality rate somewhere between 35 and 90%. So that is obviously concerning. And if you look at the mortality rates that we're seeing out of England, it is stunning. They are seeing mortality rates that are double, if not triple, what our mortality rate is per capita. So currently, as of Monday, we had reported the B117 variant, which is a variant that was first discovered in the US, in over half the states in the US, and we have reported 293 cases so far. But the thought is that this will most likely become the most dominant circulating strain of the virus by March. And most likely, as we do more surveillance, and the US is increasing how much surveillance it's doing, we will start detecting this variant elsewhere. We have unfortunately also detected one of the variants that was discovered from South Africa in a gentleman in Minnesota, and it's probable that will actually start seeing that more widely as well. There is now also this variant that we're seeing out of California. I can't remember the number that they've assigned to that, but that is another variant that they have yet to discover what its mutations are. So when we're trying to figure out, are these vaccines, are the 2 mRNA vaccines that we already have approved, are they going to be effective? Obviously, that's a $1 million question. So remember that these vaccines, actually, the mRNA expresses the RNA for the full-length spike protein, not just a receptor binding domain. So we're hoping that you generate an antibody response to both the receptor binding domain and other parts of the spike protein. And those other antibodies will still offer some protection, even if the antibody against the receptor binding domain no longer is as efficacious. So they've looked at this. So the University of Texas looked at viruses with this mutation, and they did these thing called neutralizing assays. And so they basically took cells, and then they put antibodies from people who had recovered, who had received the Pfizer vaccine, and then they added virus with this mutation, and then looked to see if the cells got infected. And with this one mutation that they saw in the B117 strain out of the UK, they did not see any of the cells get infected. So that suggested that people who've gotten vaccinated with a Pfizer vaccine will still have immunity against the strain from the US. Moderna similarly has recently released some data looking at the B117 strain from the US and the B1351 strain from South Africa, and suggested their vaccine also showed efficacy, although there was 6-fold reduced efficacy against the South African strain. Now, is that 6-fold reduced efficacy in a lab? How does that translate in the real world? We don't know that yet. Researchers at another institution actually took the sequence of the spike protein, which has 4,000 nucleotides, and mutated one nucleotide at a time to see if there was a specific nucleotide substitution that would make the antibodies from someone who's previously recovered not work anymore. And they found there were very few mutations that reduced the antibody's ability to neutralize the virus. But the one that did was this mutation called the E484 mutation. And that is unfortunately present in both the B1351 strain out of Brazil, out of South Africa, and the B11248 variant out of Brazil, but not in the B117 strain out of the UK. So that is the concerning aspect, is that is this South African strain potentially going to not be effective or evade immunity from patients who previously had COVID or patients who've been developed immunity from either the two mRNA vaccines. And we will have to wait to see. And they are going to start doing some studies. They are starting to do a phase 1 study with a modified mRNA sequence that will account for this mutation to see if that has more efficacy. But I think time will tell. If people who have previously been infected start getting infected again, and we sequence that virus and we see that it is one of these variants, then we could realize whether we are going to need to give booster shots with a slightly different form of the mRNA vaccine. So I talked about the US doing increased surveillance. So they have now established, they always had these surveillance networks, but they basically enhanced them. So they have the national SARS-CoV-2 strain surveillance called the NS3 surveillance. So this is basically using health departments who do sequence their samples and basically getting them to ship some of those samples to the CDC. And we're now the CDC is now processing 750 samples nationally a week through this. And then they're also contracting with some of the larger national reference labs to get them to do sequencing. And they're hoping, as of December 29th, they have commitments from these labs to sequence 1,700 samples a week. And then they're also contracting with universities to do genomic surveillance, so taking samples from the community and doing sequencing of those samples as well. So, we are still probably only sequencing about 1% of all our samples, and we need to do better. And hopefully with these partnerships, we will get better. And so at that point, I will stop my talk and I will stop my share, and we will see if there's any questions in the chat. I know I speak incredibly quickly. I'm sorry if I went too fast. I thought I was going to be pressed for time, trying to keep it in the 30 to 40 minute range. I seem to have gone quite quickly. So if I went too fast on any particular topic and someone wants me to go over something again, I'm more than happy to do that.
I just want to say you can put your questions in the chat or send it to Robert, which is hay@msdc.org.
Okay, so I see a question. What are your thoughts on when or if revaccinations will be needed after the second dose? So actually, Moderna is actually going to be doing they announced that they were going to be doing a study where they are probably going to give a third booster dose or a third dose to participants in the trial. Most likely it will be around July, which will be at least about 7 months after the last participant got their last dose. And they will most likely give some of the participants the same booster, so the same vaccine that they got in the first 2 doses. And some participants, they may give a slightly altered mRNA sequence booster, maybe to account for some of these variants. So this just got announced yesterday. So there's nothing in writing yet, but they already are acutely aware that they may need to give booster doses to account for some of these variants and also to maybe account for waning immunity. So far, we know that at least 3 months out, the antibody levels have been maintained at the same level that people got 1 month out. So we at least know that there's good immunity level for at least 3 months, and we will know relatively soon whether that's 5, 6 months. And the immunity that we see from the vaccines has actually been higher than the antibody level that people got from natural infection. So we're pretty confident that the immunity will probably last for quite a while from the first 2 doses of the vaccine. So the second question, when will studies begin on vulnerable populations, children, pregnant women? So actually, Pfizer's been doing the study on children for quite a while. They actually did 2 modifications. So they had the first modification where they they went down, they actually increased their sample size from 30,000 to 45,000, so they could include children 16 years and older. And they've now actually got a new study where they're actually enrolling children 12 years and older. Moderna also started up a study in December, enrolling 3,000 children aged 12 to 17. So that is actually ongoing. I do not know what the enrollment status of that is. I don't know if that's fully enrolled yet. I don't know of any study that's actively enrolling pregnant women right now. And so unfortunately, that may be a population that does not get included in these studies. You know, the current guidance from the CDC is there have been no safety concerns so far, but obviously we don't have the data. We have the 13 people in the study that got pregnant on the study, but none of those have gone to term yet. So we obviously, and even then, it will be very limited data. So right now, pregnant women are being advised to just have a conversation with the OBs. Most of the OBs I've talked to are encouraging their patients to get the vaccine because obviously the concerns about COVID in pregnancy. Surprisingly, when I looked at the data for a number of pregnant women that have been reported with COVID, it's only 55,000 patients so far. So it's less than I thought. And only 66 pregnant women have died up till now from COVID, according to the CDC data, which is less than I expected because obviously in influenza, pregnant women do much, much worse and account for a much higher proportion of the mortality based than the general population. But we may never have study data to give us clear data whether, you know, on the safety in pregnancy. So it's going to be a decision. ACOG has recommended that pregnant women do get vaccinated based on just the general safety data from the studies. Next question, person with a history of hypersensitivity with chloramphenicol giving generalized lymph node enlargement in urticaria, should he go for the vaccine? That would not be a contraindication. If there was a concern, they could wait for the J&J vaccine. I don't know, I have not seen the safety data on the J&J vaccine, but there won't be the polyethylene glycol issue with the mRNA vaccines. But there are no reasons and no contraindications to that person getting either the mRNA vaccines right now. And so if they are eligible, it may be a month or two before the J&J vaccine is widely available. So if they're a person at risk and has other, you know, high risk health conditions, I would probably recommend that they do go ahead and get vaccinated. Are there any studies in lactating new mothers? No, lactating new mothers were unfortunately excluded from the Moderna and the Pfizer vaccine. I do not know if they were included in the AstraZeneca and the J&J studies. So I don't think they were, but I don't know that for sure. Next question was, do we have recommendations about checking antibodies after patients receiving vaccines? You know, it's funny. So I got the Pfizer vaccine. I had absolutely no reaction after either dose. And I mentioned it to one of my patients. He's like, well, are you going to get the antibody level checked? And I was like, no. And he said, well, how do you know you developed immunity? And I said, you know, I guess you're right. And it just put that thought into my head because, you know, people think that, you know, having symptoms after the first or second dose is good because it suggests your immune system is reacting to the vaccine, i.e. kicking in. I'm not sure I'm going to get my antibody level checked. I trust that it works. I trust the data. And, you know, I'm not keen on having a needle stuck in my arm anyway. So I will probably just trust that the vaccine is effective. And you know, even when you get vaccinated, you have to continue using precautions, right? It's not like you're suddenly free to go sing karaoke in a bar of unmasked people. So, you know, up to this point, PPE has worked. You know, I see coronavirus patients in the hospital on a regular basis. I had not actually had a high risk exposure. I had not actually needed a coronavirus test at all until I got vaccinated, because with good PPE, you shouldn't actually have need for testing because you should be protected with that alone. So I trust that if I continue doing that, I probably won't get my antibody level checked. So I don't think it should be done regularly. I think if you have someone with absolutely no reaction, maybe it's a reason to consider checking it just to make sure they developed an immune response. So those are the only questions that I have up till now.
We have one more question. I just need to cut and paste.
Okay.
1 second. Well, actually, let me just read it out loud.
Okay.
I know of teachers and others who have tested positive for COVID after receiving the first dose. How protective is the vaccine in the real world after the first dose?
Okay.
Are the plans for teachers and other school workers to resume in-person learning after receiving only one dose congruent with existing evidence on vaccine protection?
So the data we have is obviously based on the data. So they checked, they looked at patients who developed COVID between the first and second dose of the vaccine in both the Pfizer and the Moderna. And remember, I mentioned Pfizer only had a 3-week gap between the two. And there were some cases, there were less cases in the patients who got the first dose of the Pfizer vaccine versus those who got the placebo. And they reduced the incidence of symptomatic COVID disease by 50%. In Moderna, they had a 4-week gap between the first and second. And so it reduced the incidence of symptomatic COVID by 80%. So there is definitely efficacy after the first dose. What we don't know, unfortunately, because we don't have that data, is just because you don't have symptoms, could you have been infected and we just didn't pick it up because we weren't testing everybody who wasn't developing symptoms? And it's possible that there were people who got infected that we never knew because they had asymptomatic infection. I think that the data is good enough that one dose is most likely going to give you enough protection that I think that you could say that you could open up schools with teachers only having the first of the 2 doses and not wait until every teacher has 2 doses of vaccine. I do think it is a public health urgency to get our kids back to school. I do think there has been a lot of anxiety and other, hunger issues and abuse issues that have happened because kids have not been in school. So I would like to see us try and figure out how to get kids back in school. I think there was a recent article either yesterday or today in the Washington Post suggesting that schools with social distancing, with masks, are really quite safe. And a lot of parents can't work if their kids are at home, and even when their kids are at home, they can't work because their kids need attention. And so I'm a big proponent of getting at least the youngest kids back to school. I mean, my kids are both in high school. They are doing, but they're both old enough that they're independent, and my wife can still work, and I can still go to work. But people with young kids, I think it's imperative that we try and figure out a way of getting those kids back to school. And if it means just getting teachers a single dose of the vaccine, I think that would still be okay. Any data for patients age 65 with AstraZeneca? Sorry, I'm not sure. So any data for persons age over 65 with the AstraZeneca vaccine do have antibody production data. So the data, the only data we have so far for the AstraZeneca is the data that came out of England. And I would say that is, it's muddled, and I don't think we can base too much on that yet. The US AstraZeneca study with 30,000 patients with a 2-to-1 randomization with 2 doses, 1 month apart, I think will give us that data. I just don't know. I don't have the inside scoop on the AstraZeneca data right now, but I don't think you're going to have to wait long. I guarantee you that it's in the next week or 2 that that data will be released. So I think we will have that data soon. Is it myth or science with data about school children don't get infected being in school? I mean, there definitely have been outbreaks. I mean, I think you see reports. I think that the issue is that those kids who've gotten infected have not led to outbreaks in their community. So there have been isolated outbreaks in schools. An outbreak is usually a cluster of 3 infections that are all kind of related to that, to each other. Most of those have been teachers that have actually exposed the kids, but there's been no data that any of the kids who got infected led to a wide outbreak in the community. And so I think that's why it's deemed relatively safe. It's not a high rate of outbreaks, and those outbreaks don't seem to lead to a larger community-based outbreaks. And that's why I think it's deemed relatively safe to get kids back to school. Children don't get infected, but parents likely to transmit infection. So I'm not sure if that's a statement or a question. Will there be a need to revaccinate after a year? I think most people are thinking that this is probably going to become an annual thing, similar to the flu, which is, unfortunate. But, I mean, I didn't live in 1918, but I'm sure they were facing the same questions in 1918 when they were dealing with the flu pandemic. And, now we have come to terms with living with a seasonal flu every year, except this year where we have yet to see seasonal flu. But, maybe coronavirus, supplants the seasonal flu, and instead of seasonal flu, we have seasonal coronavirus. I think we will be able to get to terms with a point where, we may need a yearly vaccination similar to what we get with the flu vaccine, but hopefully by then it will be much more widely available, and you'll be getting it at your CVS like you get your flu vaccine or at your doctor's office. And so even if there is a need, I don't think it will be a change from what we've been doing anyway. And going just as far as the flu, I think what has been remarkable is that there has been almost no flu anywhere. There at GW, we do surveillance and we test for people for the flu, and people that come in upper respiratory symptoms, we test them for COVID and we test them for other viruses. I mean, we have yet to actually diagnose a single flu case this year. And that is just unheard of. And, it's a combination of, social distancing, masks. People obviously have been getting flu vaccines, so there's that vaccination. What's interesting is that Australia is now seeing, so Australia closed its borders and got its coronavirus cases under control. And I don't know if they've relaxed their social distancing and their masks, but they are now starting to see flu outbreaks in their kids. And there's been a 6-fold increase in the incidence of flu cases in their kids at a time when you normally don't see flu in kids. So it's their summer. So obviously they normally see their flu cases in our summer, their winter, but they didn't see it when they had the initial lockdown. But now I think they've probably released some of their social distancing and some of their mask mandates because they don't have any circulating coronavirus. And now they're starting to see the flu again. So we may see flu, but we may just see it in the spring and early summer rather than the winter when we normally expect to see it. But when people were talking about worst case scenarios, it was a duemic, right? It was the flu and coronavirus on top of each other, and that would be unsustainable for our healthcare system. So luckily, we have not seen a flu pandemic on top of the coronavirus pandemic, and I think we can be very grateful for that. And I think that is it.
Well, I guess one more.
Oh, yes. Would it be possible to access slides of presentation? I can share them with Dr. Hayes, and he can do with them as he sees fit.
Yeah, it's there's going to be a video. They can go to the MSDC website, and this is being recorded. So your full presentation will be recorded.
Okay, fair enough. If I'm happy to share my slides. They're not proprietary, so I don't mind if people use my slides for their own presentations.
Yeah, thanking you, and I'm gonna also use that to thank you for your excellent and very comprehensive and up to date minutes.
The problem with coronavirus is it changes day by day. I mean, literally, I was changing some of the numbers an hour ago, and if you wait, I got to give this talk again in a week, and I'll probably have to give a totally new talk because so much will have changed and so much new information will have come out. So it is really difficult to keep an up-to-date talk on this topic.